Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. In this video we will discuss about the female gametophyte of angiosperm and its development. So at first the male reproductive part is called androecium and individual unit of it is called stamen, and female reproductive part of a flower is called as gynoecium and individual unit of the gynoecium is called as carpel. Okay, next, the pistil or female reproductive part consists of three individual structures, which is stigma, style and ovary. So it looks like this. It is the external morphology of your female reproductive part or one carpel, and it is the internal structure of carpel. Here it is called as stigma. It is the receptive part which receive the male gametes during pollination for fertilization. Then style or stylar canal inside that. Then a basal solen ovary. Inside that ovary, ovules are present. Actually, inside that ovary, many chambers are present, like the rooms of our home. So inside that locule or room, ovules are attached, and the point of attachment is called as placenta, and the pattern of arrangement of ovule inside the locule called as placentation. So female gametophyte or embryo sac of the plant will be developed inside that ovule. Within the ovary at the base of the female reproductive part. Inside the female gametophyte, generally there are 8 haploid cells, 1 egg cell, 2 synergids flanking the egg at micropyle end, then 2 polar nuclei in the center of the embryo sac, 3 antipodal cell at the chalazal end. So whole ovule have 3 ends, 1 micropyle end and 1 chalazal end. It have 2 ends, 1 micropyle end, 1 chalazal end. It is the ovule. So there are two, generally in case of angiosperms, there are 2 protective covering called integuments. Inside the integuments, nucellus is present. Inside the nucellus, embryo sac or female gametophyte is present. Inside the female gametophyte, in case of angiosperm, 8 haploid nucleus are present, 3 towards the chalazal end called as antipodal cells, 3 towards the micropyle end, which are 1 egg cell and 2 synergids, and 2 haploid nucleus at the center called as polar nuclei. Okay, next, there are different patterns of ovule attachment. One is apical placentation, where the placenta is at the apex or top of the ovary, like this. One is axial placentation, where the ovary is divided into radial segments like this, and ovules are attached to the center. This type of axial placentation is found in hibiscus, citrus and solanum. Then basal placentation, where the ovule is attached to the base of the ovary or placenta. Then free central placentation, where these ovules are attached to the center, but these ventral suture or these spokes are totally absent in case of free central placentation. Then marginal placentation, it is the simplest type of placentation. There is only one elongated placenta on one side of the ovary, and the ovules are attached at the fusion line of carpel's margin as present in Pisum sativum. Okay, P. In case of P, these are attached like this to the margin. Then parietal placentation, placenta on the inner ovary wall within a non-sextant ovary corresponding to fused carpel margin. There are 2 or more carpels are present like this. Parietal placentation generally seen in Brassicaceae family. Then superficial, similar to axial blood, but placenta are on inner surface of multilocular ovary present in nymphia. Superficial type of placentation is seen in nymphia, Nymphaceae family. Next, parts of ovule. So there is one or two protective covering called integument. Then it encloses a parenchymatous tissue called nucellus. Integument do not enclose the nucellus completely, but it have a opening called as micropyle, which allows to enter the pollen grains inside the ovule for fertilization. So opposite to micropyle, chalazal end is present. Okay, next, nucellus is the inner structure to integument. Then inside the nucellus, embryo sac is present. In immature ovule, the nucellus contains megasporocyte or megaspore mother cell, which undergoes sporogenesis via meiosis. So in that nucellus, megaspore mother cell is present, which undergoes meiosis or reductional division to form megaspores. So there are 6 type of ovules. One is orthotropous ovule or straight ovule, which is present in gymnosperm, where the body of the ovule or micropylar end, chalazal end and hilum or stalk of that ovule is present in a straight line. Then anatropous ovule, which is inverted ovule, which is present generally in all groups of angiosperms. Then hemitropous or hemianatropous ovule, where ovule is placed at right angle to the funicle or stalk of that ovule. Campylotropous, where the ovule is campylotropous ovule, which is slightly curved. Then amphitropous, which is horseshoe shaped ovule. Next is sarcinotropous ovule, where the nucellus and axis of that ovule are in same line in the beginning, but due to rapid growth on one side, the ovule becomes anatropous. The curvature continues further and micropyle again points upward. It is completely curved, okay, which is present in Cactaceae family, Nocpunsia. So these are the structure. It is the orthotropous, which is where the chalazal end, micropylar end and the stalk or funicle present in a straight line. It is the inverted ovule or anatropous. Here the stalk and the body of the ovule present in right angle, called as hemitropous or hemianatropous. Then curved ovule is called campylotropous. Then horseshoe shaped ovule, where the body of the ovule is curved as well as the embryo sac is curved, is called as amphitropous or horseshoe shaped ovule. Then where the ovule is completely curved, it is called as sarcinotropous, which is present in cactus. Next, then the female gametophyte development occur in 2 phases: megasporogenesis, megagametogenesis. The ovule develops as multicellular placental outgrowth, including epidermal and some hypodermal cell. Megaspore mother cell, actually in nucellus, some hypodermal cell become specialized to form archaeosporial cell. This archaeosporial cell divide to form sporogenous cell and primary parietal cell. Then this sporogenous cell function as megaspore mother cell. Then this megaspore mother cell undergoes meiosis to form haploid megaspores in the process of megasporogenesis. So generally from one diploid megaspore mother cell, 4 megaspores are present. 4 haploid megaspores will be developed. From these megaspores, only one towards the chalazal end, only one megaspore which is present at the chalazal end will remain functional, and other 3 will be degenerated gradually. The functional megaspore will form female gametophyte, that is embryo sac. So from megaspore mother cell, by the process of megasporogenesis, 4 haploid megaspores are formed. From that 4 haploid megaspores, 3 will be degenerated, and one megaspore which is present towards the chalazal end will remain functional and forms the whole female gametophyte or embryo sac by the process of megagametogenesis. Next. Then the megaspore is the first cell of female gametophyte. It becomes enlarged at the expense of some nutrition and the nucellus, and thus form female gametophyte. Initially embryo sac is uninucleate, and with further growth its nucleus divide by 3 successive divisions to form 8 nuclei. So this embryo sac divide 3 successive divisions to form 8 nuclei. From these 8 nuclei, 3 moves towards the chalazal end, 3 moves towards the micropylar end, and 2 remain at the center. Okay, at the micropylar end 2 synergids and 1 egg cell is present. At the chalazal end 3 antipodal cells are present. At the center, polar nuclei are present inside a large cell. So this type of development of embryo sac is called as polygonum type of embryo sac. Monosporic and polygonum. Monosporic means where generally from out of 4 resultant megaspore, 1 will remain functional and form the whole female gametophyte. That type of embryo sac development is called as monosporic type of embryo sac development, and typically this type of development is called monosporic polygonum type of development. So there are other types of embryo sac development like bisporic and tetrasporic. In case of bisporic, two nucleus, 2 cell will remain functional and form the whole female gametophyte. In case of tetrasporic type of female gametophyte development, all the 4 cells will remain functional and form the whole embryo sac. In case of monosporic, 1 functional megaspore will form the whole female gametophyte or embryo sac. In case of bisporic embryo sac, 2 nucleus or 2 megaspores will remain functional and form the female gametophyte. In case of tetrasporic type of female gametophyte development, 4 functional megaspores will form the female gametophyte. So this type of embryo sac development will further divided into some subcategories. Okay, so I think I should end this lecture video here. In my next video, I will discuss the types of monosporic, bisporic and tetrasporic embryo sac development, and that will be the part 2 video of this female gametophyte and its development video. So please stay tuned and enjoy my videos like this. I hope you really get benefit from these videos, and please like it, please share with your other friends, teachers, family members, and please subscribe to my channel Sunanda's Tutorial. Thank you.